Chikungunya is an infection caused by the chikungunya virus. Symptoms include the sudden onset of fever 2 to 4 days after exposure. The fever usually lasts 2 to 7 days, while accompanying joint pains typically last weeks or months but sometimes years. The risk of death is a little less than 1 in 1,000. The elderly or those with underlying chronic medical problems are most likely to have severe complications. The virus is passed to humans by two species of mosquito of the genus Aedes, A. albopictus and A. aegypti. The virus circulates within a number of animals, including monkeys, birds, cattle, and rodents. This is in contrast to dengue, for which primates are the only ones affected. The best means of prevention is overall mosquito control and the avoidance of bites by mosquitoes in areas where the disease is common. This may be partly achieved with the use of mosquito nets. No specific treatment is known, but supportive care is recommended, including rest, fluids, and medications to reduce fever and joint pain. Since 2004, the disease has occurred in outbreaks in Asia, Europe, and the Americas. Signs and symptoms. The incubation period of the chikungunya virus ranges from 1 to 12 days, and is most typically 3 to 7. The disease may be asymptomatic, but generally is not, as 72% to 97% of those infected will develop symptoms. Characteristic symptoms include sudden onset with high fever, joint pain, and rash. Other symptoms may occur, including headache, fatigue, digestive complaints, and conjunctivitis. Information gained during recent epidemics suggests that chikungunya fever may result in a chronic phase as well as the phase of acute illness. Within the acute phase, two stages have been identified. A viral stage during the first 5 to 7 days, during which viremia occurs, followed by a convalescent stage lasting approximately 10 days, during which symptoms improve and the virus cannot be detected in the blood. Typically, the disease begins with a sudden high fever that lasts from a few days to a week, and sometimes up to 10 days. The fever is usually above 39 degrees Celsius and sometimes reaching 40 degrees Celsius and may be biphasic, lasting several days, breaking, and then returning. Fever occurs with the onset of viremia, and the level of virus in the blood correlates with the intensity of symptoms in the acute phase. When IgM, an antibody that is a response to the initial exposure to an antigen, appears in the blood, viremia begins to diminish. However, headache, insomnia, and an extreme degree of exhaustion remain, usually about 5 to 7 days. Following the fever, strong joint pain or stiffness occurs. It usually lasts weeks or months, but may last for years. The joint pain can be debilitating, or often resulting in near immobility of the affected joints. Joint pain is reported in 87 to 98% of cases, and nearly always occurs in more than one joint, though joint swelling is uncommon. Typically, the affected joints are located in both arms and legs, and are affected symmetrically. Joints are more likely to be affected if they have previously been damaged by disorders such as arthritis. Pain most commonly occurs in peripheral joints, such as the wrists, ankles, and joints of the hands and feet, as well as some of the larger joints, typically the shoulders, elbows, and knees. Pain may also occur in the muscles or ligaments. Rash occurs in 40 to 50% of cases, generally as a maculopapular rash occurring 2 to 5 days after onset of symptoms. Digestive symptoms, including abdominal pain, nausea, vomiting or diarrhea, may also occur. In more than half of cases, normal activity is limited by significant fatigue and pain. Infrequently, inflammation of the eyes may occur in the form of iridocyclitis or uveitis, and retinal lesions may occur. Rarely, neurological disorders have been reported in association with chikungunya virus, including Guillain-Barré syndrome, palsies, meningoencephalitis, flaccid paralysis, and neuropathy. In contrast to dengue fever, chikungunya fever very rarely causes hemorrhagic complications. Symptoms of bleeding should lead to consideration of alternative diagnoses: a co-infection with dengue fever, a coexisting congestive hepatopathy. Chronic disease observations during recent epidemics have suggested chikungunya may cause long-term symptoms following acute infection. This condition has been termed chronic chikungunya virus-induced arthralgia. Long-term symptoms are not an entirely new observation. Long-term arthritis was observed following an outbreak in 1979. Common predictors of prolonged symptoms are increased age and prior rheumatological disease. During the La Réunion outbreak in 2006, more than 50% of subjects over the age of 45 reported long-term musculoskeletal pain, with up to 60% of people reporting prolonged painful joints 3 years following initial infection. A study of imported cases in France reported that 59% of people still suffered from arthralgia 2 years after acute infection. Following a local epidemic of chikungunya in Italy, 66% of people reported muscle pains, joint pains, or asthenia at 1 year after acute infection. Currently, the cause of these chronic symptoms is not fully known. Markers of autoimmune or rheumatoid disease have not been found in people reporting chronic symptoms. However, some evidence from humans and animal models suggests chikungunya may be able to establish chronic infections within the host. Viral antigen was detected in a muscle biopsy of a person suffering a recurrent episode of disease 3 months after initial onset. Additionally, viral antigen and viral RNA were found in macrophages in the synovial joint of a person experiencing a relapse of musculoskeletal disease 18 months after initial infection. Several animal models have also suggested chikungunya virus may establish persistent infections. In a mouse model, viral RNA was detected specifically in joint-associated tissue for at least 16 weeks after inoculation, and was associated with chronic synovitis. Similarly, another study reported detection of a viral reporter gene in joint tissue of mice for weeks after inoculation. In a non-human primate model, chikungunya virus was found to persist in the spleen for at least 6 weeks. Cause: Virology. Chikungunya virus, also referred to as CHIKV, is a member of the alphavirus genus and Togaviridae family. It is an RNA virus with a positive sense single-stranded genome of about 11.6 kilobase. It is a member of the Semliki forest virus complex and is closely related to Ross River virus, Onyong virus, and Semliki forest virus. Because it is transmitted by arthropods, namely mosquitoes, it can also be referred to as an arbovirus. In the United States, it is classified as a Category C priority pathogen, and work requires biosafety Level 3 precautions. Transmission: chikungunya is generally transmitted from mosquitoes to humans. Less common modes of transmission include vertical transmission, which is transmission from mother to child during pregnancy or at birth. Transmission via infected blood products and through organ donation is also theoretically possible during times of outbreak, though no cases have yet been documented. Chikungunya is related to mosquitoes, their environments, and human behavior. The adaptation of mosquitoes to the changing climate of North Africa around 5,000 years ago made them seek out environments where humans stored water. Human habitation and the mosquitoes' environments were then very closely connected. During periods of epidemics, humans are the reservoir of the virus, because high amounts of virus are present in the blood in the beginning of acute infection. The virus can be spread from a viremic human to a mosquito, and back to a human. During other times, monkeys, birds, and other vertebrates have served as reservoirs. 3 genotypes of this virus have been described, each with a distinct genotype and antigenic character. West African, East, Central, South African, and Asian genotypes. Chikungunya is spread through bites from Aedes mosquitoes, and the species A. aegypti was identified as the most common vector, though the virus has recently been associated with many other species, including A. albopictus. Research by the Pasteur Institute in Paris has suggested chikungunya virus strains in the 2005-2006 Reunion Island outbreak incurred a mutation that facilitated transmission by the Asian tiger mosquito. Other species potentially able to transmit the chikungunya virus include A. furcifatela, A. africanus, and A. luteocephalus. Mechanism: the chikungunya virus is passed to humans when a bite from an infected mosquito breaks the skin and introduces the virus into the body. The pathogenesis of chikungunya infection in humans is still poorly understood, despite recent outbreaks. It appears that in vitro, chikungunya virus is able to replicate in human epithelial and endothelial cells, primary fibroblasts, and monocyte-derived macrophages. Viral replication is highly cytopathic, but susceptible to type 1 and 2 interferon. In vivo, in studies using living cells, chikungunya virus appears to replicate in fibroblasts, skeletal muscle progenitor cells, and myofibers. The type 1 interferon response seems to play an important role in the host's response to chikungunya infection. Upon infection with chikungunya, the host's fibroblasts produce type 1 alpha and beta interferon. In mouse studies, deficiencies in INF1 in mice exposed to the virus cause increased morbidity and mortality. The chikungunya-specific upstream components of the type 1 interferon pathway involved in the host's response to chikungunya infection are still unknown. Nonetheless, mouse studies suggest that IPS1 is an important factor, and that IRF3 and IRF7 are important in an age-dependent manner. Mouse studies also suggest that chikungunya evades host defenses and counters the type 1 interferon response by producing NS2, a non-structural protein that degrades RBP1 and turns off the host cell's ability to transcribe DNA. NS2 interferes with the JAK-STAT signaling pathway and prevents STAT from becoming phosphorylated. In the acute phase of chikungunya, the virus is typically present in the areas where symptoms present, specifically skeletal muscles and joints. In the chronic phase, it is suggested that viral persistence, lack of clearance of the antigen, or both contribute to joint pain. The inflammation response during both the acute and chronic phase of the disease results in part from interactions between the virus and monocytes and macrophages. Chikungunya virus disease in humans is associated with elevated serum levels of specific cytokines and chemokines. High levels of specific cytokines have been linked to more severe acute disease. Interleukin-6, IL-1 beta, RANTES, monocyte chemoattractant protein 1, monokine induced by gamma interferon, and interferon gamma induced protein 10. Cytokines may also contribute to chronic chikungunya virus disease, as persistent joint pain has been associated with elevated levels of IL-6 and granulocyte macrophage colony stimulating factor. In those with chronic symptoms, a mild elevation of C-reactive protein has been observed, suggesting ongoing chronic inflammation. However, there is little evidence linking chronic chikungunya virus disease and the development of autoimmunity. Viral replication. The virus consists of 4 non-structural proteins and 3 structural proteins. The structural proteins are the capsid and 2 envelope glycoproteins, E1 and E2, which form heterodimeric spikes on the virion surface. E2 binds to cellular receptors in order to enter the host cell through receptor-mediated endocytosis. E1 contains a fusion peptide which, when exposed to the acidity of the endosome in eukaryotic cells, dissociates from E2 and initiates membrane fusion that allows the release of nucleocapsids into the host cytoplasm, promoting infection. The mature virion contains 240 heterodimeric spikes of E2, E1, which after release bind on the surface of the infected cell, where they are released by exocytosis to infect other cells. Diagnosis. Chikungunya is diagnosed on the basis of clinical, epidemiological, and laboratory criteria. Clinically, acute onset of high fever and severe joint pain would lead to suspicion of chikungunya. Epidemiological criteria consist of whether the individual has traveled or spent time in an area in which chikungunya is present within the last 12 days. Laboratory criteria include a decreased lymphocyte count consistent with viremia. However, a definitive laboratory diagnosis can be accomplished through viral isolation, RT-PCR, or serological diagnosis. The differential diagnosis may include infection with other mosquito-borne viruses, such as dengue or malaria, and infection with influenza. Chronic recurrent polyarthralgia occurs in at least 20% of chikungunya patients 1 year after infection, whereas such symptoms are uncommon in dengue. Virus isolation provides the most definitive diagnosis, but takes 1 to 2 weeks for completion and must be carried out in biosafety level 3 laboratories. The technique involves exposing specific cell lines to samples from whole blood and identifying chikungunya virus-specific responses. RT-PCR using nested primer pairs is used to amplify several chikungunya-specific genes from whole blood, generating thousands to millions of copies of the genes in order to identify them. RT-PCR can also be used to quantify the viral load in the blood. Using RT-PCR, diagnostic results can be available in 1 to 2 days. Serological diagnosis requires a larger amount of blood than the other methods, and uses an ELISA assay to measure chikungunya-specific IgM levels in the blood serum. One advantage offered by serological diagnosis is that serum IgM is detectable from 5 days to months after the onset of symptoms, but drawbacks are that results may require 2 to 3 days, and false positives can occur with infection due to other related viruses, such as Onyong virus and Semliki forest virus. Presently, there is no specific way to test for chronic signs and symptoms associated with chikungunya fever, although non-specific laboratory findings such as C-reactive protein and elevated cytokines can correlate with disease activity. Prevention. Because no approved vaccine exists, the most effective means of prevention are protection against contact with the disease-carrying mosquitoes and controlling mosquito populations by limiting the habitat. Mosquito control focuses on eliminating the standing water where mosquitoes lay eggs and develop as larvae. If elimination of the standing water water is not possible. Insecticides or biological control agents can be added. Methods of protection against contact with mosquitoes include using insect repellents with substances such as DEET, picaridine, PMD, or IR3535. However, increasing insecticide resistance presents a challenge to chemical control methods. Wearing bite-proof long sleeves and trousers also offers protection, and garments can be treated with pyrethroids, a class of insecticides that often has repellent properties. Vaporized pyrethroids are also insect repellents, as infected mosquitoes often feed and rest inside homes. Securing screens on windows and doors will help to keep mosquitoes out of the house. In the case of the day-active A. aegypti and A. albopictus, however, this will have only a limited effect, since many contacts between the mosquitoes and humans occur outdoors. The vaccine currently, no approved vaccines are available. A Phase 2 vaccine trial used a live, attenuated virus to develop viral resistance in 98% of those tested after 28 days, and 85% still showed resistance after 1 year. However, 8% of people reported transient joint pain, and attenuation was found to be due to only 2 mutations in the E2 glycoprotein. Alternative vaccine strategies have been developed and show efficacy in mouse models. In August 2014, researchers at the National Institute of Allergy and Infectious Diseases in the USA were testing an experimental vaccine which uses virus-like particles instead of attenuated virus. All the 25 people participated in this Phase 1 trial developed strong immune responses. Phase 2 trial will commence using 400 adults aged 18 to 60 and take place at 6 locations in the Caribbean. Even with a vaccine, mosquito population control and bite prevention will be necessary to control chikungunya disease. Treatment. Currently, no specific treatment for chikungunya is available. Supportive care is recommended, and symptomatic treatment of fever and joint swelling includes the use of non-steroidal anti-inflammatory drugs such as naproxen, non-aspirin analgesics such as paracetamol, and fluids. Aspirin is not recommended due to the increased risk of bleeding. Despite anti-inflammatory effects, corticosteroids are not recommended during the acute phase of disease, as they may cause immunosuppression and worsen infection. Passive immunotherapy has potential benefit in treatment of chikungunya. Studies in animals using passive immunotherapy have been effective, and clinical studies using passive immunotherapy in those particularly vulnerable to severe infection are currently in progress. Passive immunotherapy involves administration of anti-CHIKV hyperimmune human intravenous antibodies to those exposed to a high risk of chikungunya infection. No antiviral treatment for chikungunya virus is currently available, though testing has shown several medications to be effective in vitro. Chronic arthritis in those who have more than 2 weeks of arthritis, ribavirin may be useful. The effect of chloroquine is not clear. It does not appear to help acute disease, but tentative evidence indicates it might help those with chronic arthritis. Steroids do not appear to be an effective treatment. NSAIDs and simple analgesics can be used to provide partial symptom relief in most cases. Methotrexate, a drug used in the treatment of rheumatoid arthritis, has been shown to have benefit in treating inflammatory polyarthritis resulting from chikungunya, though the drug mechanism for improving viral arthritis is unclear. Prognosis. The mortality rate of chikungunya is slightly less than 1 in 1,000. those over the age of 65, neonates, and those with underlying chronic medical problems are most likely to have severe complications. Neonates are vulnerable as it is possible to vertically transmit chikungunya from mother to infant during delivery, which results in high rates of morbidity as infants lack fully developed immune systems. The likelihood of prolonged symptoms of chronic joint pain is increased with increased age and prior rheumatological disease. Epidemiology. Historically, chikungunya has been present mostly in the developing world. The disease causes an estimated 3 million infections each year. Epidemics in the Indian Ocean, Pacific Islands, and in the Americas continue to change the distribution of the disease. In Africa, chikungunya is spread by a silvertich cycle in which the virus largely cycles between other non-human primates, small mammals, and mosquitoes between human outbreaks. During outbreaks, due to the high concentration of virus in the blood of those in the acute phase of infection, the virus can circulate from humans to mosquitoes and back to humans. The transmission of the pathogen between humans and mosquitoes that exist in urban environments was established on multiple occasions from strains occurring on the eastern half of Africa in non-human primate hosts. This emergence and spread beyond Africa may have started as early as the 18th century. Currently available data does not indicate whether the introduction of chikungunya into Asia occurred in the 19th century or more recently, but this epidemic Asian strain causes outbreaks in India and continues to circulate in Southeast Asia. In Africa, outbreaks were typically tied to heavy rainfall causing increased mosquito population. In recent outbreaks in urban centers, the virus has spread by circulating between humans and mosquitoes. Global rates of chikungunya infection are variable, depending on outbreaks. When chikungunya was first identified in 1952, it had a low-level circulation in West Africa, with infection rates linked to rainfall. Beginning in the 1960s, periodic outbreaks were documented in Asia and Africa. However, since 2005, following several decades of relative inactivity, chikungunya has re-emerged and caused large outbreaks in Africa, Asia, and the Americas. In India, for instance, chikungunya reappeared following 32 years of absence of viral activity. Outbreaks have occurred in Europe, the Caribbean and South America, areas in which chikungunya was not previously transmitted. Local transmission has also occurred in the United States and Australia, countries in which the virus was previously unknown. In 2005, an outbreak on the island of Acute was the largest then documented, with an estimated 266,000 cases on an island with a population of approximately 770,000. In a 2006 outbreak, India reported 1.25 million suspected cases. Chikungunya was recently introduced to the Americas, and from 2013-14 in the Americas, 1,118,763 suspected cases and 24,682 confirmed cases were reported by the PAHO. An analysis of the chikungunya virus's genetic code suggests that the increased severity of the 2005 present outbreak may be due to a change in the genetic sequence which altered the E1 segment of the virus's viral coat protein, a variant called E1A226V. This mutation potentially allows the virus to multiply more easily in mosquito cells. The change allows the virus to use the Asian tiger mosquito as a vector in addition to the more strictly tropical main vector, Aedes aegypti. Enhanced transmission of chikungunya virus by A. albopictus could mean an increased risk for outbreaks in other areas where the Asian tiger mosquito is present. A. albopictus is an invasive species which has spread through Europe, the Americas, the Caribbean, Africa, and the Middle East. After the detection of Zika virus in Brazil in April 2015, the first ever in the Western Hemisphere, it is now thought some chikungunya and dengue cases could in fact be Zika virus cases or co-infections. History. The word chikungunya is believed to have been derived from a description in the Makonde language meaning that which bends up, of the contorted posture of people affected with the severe joint pain and arthritic symptoms associated with this disease. The disease was first described by Marion Robinson and W. H. R. Lumsden in 1955, following an outbreak in 1952 on the Makonde Plateau, along the border between Mozambique and Tanganyika. According to the initial 1955 report about the epidemiology of the disease, the term chikungunya is derived from the Makonde root verb kungunyala, meaning to dry up or become contorted. In concurrent research, Robinson glossed the Makonde term more specifically as that which bends up. Subsequent authors apparently overlooked the references to the Makonde language and assumed the term to have been derived from Swahili, the lingua franca of the region. The erroneous attribution to Swahili has been repeated in numerous print sources. Many erroneous spellings of the name of the disease are also in common use. Since its discovery in Tanganyika, Africa, in 1952, chikungunya virus outbreaks have occurred occasionally in Africa, South Asia, and Southeast Asia, but recent outbreaks have spread the disease over a wider range. The first recorded outbreak of this disease may have been in 1779. This is in agreement with the molecular genetics evidence that suggests it evolved around the year 1700. Biological weapon. Chikungunya was one of more than a dozen agents researched as potential biological weapons. Research. Infection was reduced in mosquitoes with the WMEL strain of Wolbachia.